Right. Thank you very much. So thank you very much, the organizer. It's a great pleasure to be here. Given that I have very little time and probably much too many slides, let me just try this one out here. Yep. Let me get into it right away. Here's my conflict of interest slide. So what I will be talking about today is biochemical fingerprint of disease. And the question I get very quickly through the motivation, I think we've seen that already several times today. So what really makes us different? What's the cause of complex outcomes? It's, of course, our lifestyle, how we live, how we behave, are we physically active, what drugs do we take? And then, of course, I mean, we haven't heard much about this today, the gut microbiome, and then very centrally, of course, our own genome. So all these factors lead to complex disorders, and what we really want to understand is how do these come together? And I think the point I want to make today is, I'm talking about metabolomics, is if you take the paradigm of biology, you come from the gene to the transcriptome to the proteomes, you can do all this omics analysis, and there's also modifications, DNA modification, microRNAs, protein modifications like comics. All these are actually things downstream of the genome, but if you really come to the intermediate phenotype that actually comes where the biology happens, that's the metabolomics. That's what's really circulating. So the idea is actually you have in my point of view, there is this biochemical factory. I mean, I think there are 600 factories sitting around here. The genome, the proteins make up this factory, but what really goes through the factory is the metabolome. And that's really what the phenotype is we're interested in metabolomics. And also here very quickly, so metabolomics for me is just this kind of pathway networks where each dot is a metabolite and they are connected through the enzymes, the transporters, the regulators. And what you really want to understand is get a whole picture of it. And in metabolomics, it's more complicated than in genomics. I mean, genes are all basically the same makeup. Metabolites, they're all biochemically very different, very hard to measure. And I think the other important point to anticipate, I think in here I don't have to run in too many open doors, but the idea is population studies, they're really experiments created by nature. So if you can go for 500,000 people in UK Biobank, have all the metabolites on them, and then study all these things together, it's really like doing experiments conducted by nature. So what I want to show you now is association studies. So I think that's we've seen also, like you can do association between the genes, the microbiome, PHEWAS, everything. But the challenge is, like these population studies, they identify risk factors, but they really rarely get to the bottom of it, to where the pathways are. So there's a question mark in the middle, and I think something we also have already very well understood is that all these omics are very well intermediate phenotypes. And then again, you can go and associate the genome and the phenome and everything to the disease. And which brings us to the point that actually all these deep molecular phenotypes, so I'm not advocating here to do metabolomics instead of something, but all these intermediate phenotypes actually reveal the affected pathways and can really indicate therapy options. And what I now really wanted to show you is early work we did on GWAS with metabolomics, and then from there on maybe take you a bit further and give you some of a taste of what the phenotype really gives you. So in 2008, we conducted the first GWAS with metabolomics in a rather small study, and what's really amazing is that even in this small study, we find what we now term the genetically identified metabotypes. That's a genetic association between metabolites and genetic variants. And that we have seen it repeatedly again and again. The thing is, like what you find is that there's a very strong difference between the minor allele, the major allele homozygotes. In this case, sometimes a 50% difference. And I like to see this as valves. So in the end, each of us, we have all our enzymes in our body, but then the genetic variants, they give different settings of our valves. So these associations here is not disease. This is different properties of everyone in this room. And if you take, for instance, this one variant in the FADS1 gene, it's a minor allele frequency of 20%. So I think in this room, we would have about 25 people who are actually the minor allele homozygotes who have a very different metabolism of these polyunsaturated fatty acids. But I think there are not 25 sick people here. So that's really a different setting of these valves. And then when we initially published it, I think other people actually pointed me on this. I didn't even know Archibald Garrett. I'm a physicist, but I think biochemists know him very well. He's the guy who invented or refound the inborn errors of metabolism. But he said in 1902, actually, that these are only mere examples of extreme variation. And he already realized that there's something like complex disorders. So he said chemical individuality, or now we call it more metabolic individuality, is actually predisposition to disease. And that's really what we want to study with metabolomics in studies like the UK Biobank. And that's something he did in 1909. So I bought the book from him. It's really like you read a book from 10 years ago. So once we had this, we said we want to go forward and try to map out the atlas of human metabolic individuality, trying to get all relevant genetically influenced metabotypes, and then think about, I will talk about that at the end, how to translate that to medical application. So the first step was to do metabolomics. So here you see the Helmholtz Center where I was working at the time. You need a lot of mass specs, and I think we will hear in the next talk a lot more about how to actually go about this. At that time, we decided to go especially with companies, so work with Biocritus, with Numerus, Genomics, and Metabolon, and then ran a series of GWAS. So the first one was a very slowly powered. Then we tried the Biocritus kit, which is a targeted lipidomics assay, Genomics, NMR in urine, and every time the story was the same. We had this genetically influenced metabotypes coming up with a phenotype that metabolically matched. And then I think really the breakthrough came when we hit when we met Metabolon. I think the Helmholtz Center, the Cora Accord, was the first to actually work at that scale with Metabolon. And we did a study where we ran nearly 3,000 samples together with a control here from Twins UK. And so maybe let me show you this example a bit my fascination for the whole thing. So what you see here is 37 genome-wide replicated hits, and they are 37 different stories. So each one tells one story. There's a common theme. You see FATS1, for instance, goes exactly what the protein does. It puts in the fourth double bond in the polyacylated fatty acids. You see even here nucleotidase with inosine, dipeptides with the ACE enzyme, and so on. So there's a perfect match between the phenotype, where the genes sit in the enzymes, and the metabotype. And for me, that was never something obvious, because in the end, you take a drop of blood, you run it through mass spectrometers, all kind of stuff, and you get in the end 500 metabolite values, and then you do another drop of blood, you run it through all kind of Affymetrix arrays and things like that. And then you do an association of hundreds with hundreds of thousands, and in the end, all the p-values are flat except a few, and that these few are actually SNPs located in or near the enzymes and matching their metabotype. That was far from being the case, and that's actually what we really find. So this match between gene function and associated metabotype, I think for me at that time was really a big surprise. And then knowing that, of course, you can go on. Like here, we have an association of carnitine with an orphan transporter, and we could show that this is actually a transporter. So you can use genomics data to do functional genomics. I have other examples you can do the other way around. You can also annotate unknown metabolites using genetics. I won't talk about that. Gene-environment interaction. So the FATs I showed you before, people later study genetic variant and interaction with intake of omega-6, omega-3 fatty acids, and they actually show there's an interaction between them and the outcome of LDL and cholesterol. So you can use these genetic variants to actually predict gene-environment interaction. Other things is ECAT-S. So this is a gene where we predicted an effect on beta oxidation. And this here is from an abstract where they actually cite our study, and they designed a study based on that GWAS, and they actually found, yes, this genetic variant in ECAT-S, and this is not monogenic disorder, is actually having a phenotype that's diabetes relevant. So you can then generate hypotheses for clinical trials. And then I think the point, this is from the publication in 2012. I think this is now much more dense. The thing is, when you overlap now all these different variants with the GWAS catalog, and today you would do it more sophisticatedly with colocylation and things like that, you actually find exactly what we expected, that metabolomics is really, for many, many variants, the intermediate phenotype on which you can then try to understand and interpret the GWAS hits. So interpretation of GWAS diseases, and then also, I think pharma industry is extremely interested in that, because all of these things is extra phenotype to actually support that a certain gene is a good target for a drug. So we continued, so you can work with unknown diseases, you can work with NMR, and I think the next big step was the publication that we did together here with Tim Spector, and it was funded by Pfizer. In that study we did nearly 8,000 people, 400 metabolites, all annotated by Metabolon. I think this annotation is really what made the thing really happen. What we find here now, this would be now 145 different individually genetically influenced metabotypes. And each of them it's a story on its own. And then if you just classify them, you would see there's a lot of enzymes, a lot of transporter, what I said already. Many that are actually this kind of weak variants of inborn errors. So you have a lot of the inborn errors of metabolism. You also see them as moderate variants. These are actually the valve settings I'm talking about. A lot of things overlapping with drug targets that are already known, and then they overlap with the disease. So there's really an extreme richness of how to take the metabolites as the endpoints of biology. And then you can put them together, so there's systems biology methods to do GGMs and networks and things, and connect actually in a network the metabolites to each other, the variants and the disease. So that's what we call the atlas. And then if you have this huge network, you can pull on it. So you can say, okay, I'm interested in cardiovascular. So here would be in the colors would be coronary artery disease related GWAS phenotypes, the variants here, the metabolites. And then I don't want to discuss the whole thing, but if you look at it, you see there's the whole story of a coronary artery disease. But this here is drawn from data. It's drawn from this experiment driven by nature on us by having all the metabolites on thousands of people and the genetics. And so that's for identification of pathways. And then very recently, so I'm mentoring Praveens Run, and this is a study done with Adam Butterworth and Claudia Langenberg in Cambridge. So this is unpublished yet, so can you show you a little bit, but the amazing thing is this is a study with 20,000 people, 900 metabolites, and what you see here is, like, this is colored by metabolic pathway. So each line here is a Manhattan plot. So this is the Manhattan plot of a study with 20,000. And just a brief thing, how things go now. So before we had a metabotype going with a genetic variant. Now we have enough power to actually see multiple signals, and we've gone through some lens to really bring them together. So you would see here a frequent variant associating with a lot of metabolites, some up, some down. Another frequent one, only with a few of them, and then rare variants as well. And then when you look at them, I don't have the details here, but you would see there could be variants that actually change the expression level. So all metabolites would go the same way, the substrates up, the products down. And then another variant could be a coding variant that would play on the substrate specificity. So especially if you look at this column here, everything is red, and here is a green. So there's really something very different. And now we have about, I think, 300 or so of these really mini matrices, and each of them, we annotated them, they go back, and you find biochemistry from 100 years ago. I mean, we have the literature from 100 years ago covering these 300 different loci. It's so amazing that I think the publication of the whole thing is delayed because we just can't stop looking at the data. So to wrap this part up, so there's a match between the metabotype and the gene function, surprisingly large effect sizes, frequent genotypes that are really relevant, and that's this kind of valve setting we're talking about, overlap with GWAS loci, intermediate phenotype to disease, gene-environment interaction. So let me just not stick too much with that, because I said I'm trying to speed up a bit to get you also something new and non-genetic. So the thing is, if we would now do metabolomics in the UK Biobank, of course you don't just want to do another GWAS. So you want to go further. And we did a study in 2014 where we did an EWAS, so epigenetic association with metabolomics. And to be very honest, at that point, I was disappointed. I didn't understand what we're talking about, and we'll show you why. So what we studied is an association between Illumina 450K methylation, the different metabolomics data we had in the CORA study, and did an EWAS. So just like a GWAS, you do it on the methylation. And the hypothesis is that differentially methylation is a proxy for different gene expression, and that would actually indicate what genes are playing a role and where. And what we found is, here's then the Manhattan plot for the EWAS, and actually we're a bit disappointed because many of the hits were just GWAS hits. And that's probably due to the fact that these SNPs are interacting. They may maybe increase the efficiency of an enzyme, and then the methylation just brings it back to the level you know, or something like that. But then you could go and add variants and take out the genetic variant as a covariate, and then you would still find a lot of things. So there's associations not confounded by SNPs. And you don't have to look at the thing in detail, but we found 20 different things, CPG sites, and then you could look at them. And here comes this environment into play, because there was a whole set, they were all associated with smoking. And this comes actually from a GWAS with smoking, and this all are genes that are actually playing a role in detoxification of smoking. There's another group here, and then the one which I really at the time didn't really well understand, that these are individual associations, and I just highlight 3 of them here. So that's TXNP, ABCG1, and CPT1A. So some of you might have heard that from afterwards, because they are actually hits for epigenetic association with diabetes. So here is them again. And really the take-home message is here. Before I just looked at the top metabolite, just one metabolite. And that is okay, but if you only have a metabolite, it doesn't tell you much. But if you look at the environment, the full metabotype, so we pulled these CPGs again, and then you find suddenly an association with alpha-hydroxybutyrate, with glycine, with phosphocholine, with all kind of stuff. And these have been seen before in a metabolome-wide association with diabetes. So all these three are actually diabetes phenotype, and we could have derived in 2014 that these three are diabetes risk loci for EWAS. We didn't, but later on we understood that. So the idea actually is why would the methylation go up a bit down? The thing is, the organ would, if you have 30% methylation, there would be maybe 30% of the cells would actually do something to cope with the stress, with the smoking, with the diabetes state, with whatever. If you move on in your disease, you would get more of them. So the association comes actually from the fact that the CpG methylation is a readout what the organs actually do to cope with disease. So methylation can actually be an early indicator, and having that combined with metabolomics would give you the thing. So the thing is clucking red, so I come to my conclusion. The idea is genetic predisposition is actually the predisposition factor. And these are the valves. Then you have the lifestyle choices, environment, and everything. They are the driving factors that lead to changes in all these different metabotypes and intermediate phenotypes, which then induce a reaction of the body in gene expression, and that can be read out by methylation. So the methylation is then the dials, the dial gauges. And then that may go on and again and again. So if you take metabolomics and you combine that with genetics and epigenetics, you actually get, so you actually come to this picture. So the idea is you have this biochemistry factory that we all are. You have the valves and the gauges, and now the selling point is, of course, if you could get all this data in UK Biobank, and this is really, I promise you, my last slide, and I think this one is from a last meeting we had for the enrichment thing, is you can see how many cases are actually expected in UK Biobank for different diseases. So just imagine you would have all this metabolomics data combined with all this phenotype, what you could do with that. And I think that's, with that, I would like to thank my funders, Qatar Foundation, my group in Qatar, my more extended group in the world. Thank you very much. And if you're interested in more data, I have a blog where there's all this kind of tables of GWAS and EWAS with metabolomics. Thanks a lot.